Hi, everyone. I hope everyone can see me and my screen here. This is the first time presenting on this app, but we're going to give it a shot. So I'm going to go ahead and share my screen so you can see the PowerPoint fully. And if you want to ask questions during the presentation, I think you should be able to type them into a chat, and I will answer them when I'm done with my presentation in a few minutes. All right, so I am presenting for myself and Dr. Bernadine Strick, who you just heard all about. We have been working on a project in alternative management systems in Columbia Star, looking at planting density and primocane management and how those things affect yield and labor costs. So I've just got a few slides, but I want to walk you through our results from the first harvest season of this project and talk to you about kind of what our goals are for the future as well. For treatments, we're looking at plant spacing for the first part. So the industry standard is typically to plant at 5 feet, which is what you see in the background. And then we're comparing that to a higher density planting at 2.5 feet, which is right there in the front. These plants were planted originally in fall of 2019. We did have a hard frost right after planting. And so we had to replant some of them in the spring of 2020. So this is, a few months after some of them started growing. By the next year, they had really kind of evened out and grew consistently. So you can see the really dense solid canopy at the 2.5 feet spacing on the bottom compared to the 5 foot spacing on the top, where there's a little bit more space in the canopy a year after planting. The other treatments that we're looking at are primocane suppression. So the picture on the left was in May when we did not do any primocane suppression, compared to the bottom picture where we did primocane suppression. This picture was taken a couple of weeks after our application of AIM. We just did one primocane suppression application. And the goal with this is to try and improve machine harvest efficiency. So that gets rid of some of those fruiting laterals down there. And in theory should, the catcher plate should be able to close more closely around the plant and potentially catch more of that fruit that falls. And then finally, we're looking at training methods. So doing the traditional kind of August training where we cane out all the floricanes and train the primocanes up in August. And comparing that to the new over old system where the floricanes do not get caned out. They just stay on the plant for several years. And then the new primocanes get trained up over the floricanes. So obviously this is a time-saving method, but we are concerned about disease, especially with the blackberry collapse disease that's become an issue in some varieties like Columbia Star and Black Diamond. So we'll be keeping an eye out for that. We haven't seen any signs of it yet, but I know that can take several years potentially to get going in a field. So we will be keeping an eye out for those things. So this is sort of just a photo montage of the 2021 growing season. We machine harvested these plants 4 times this year. Obviously, we were hit by the heat dome just like everybody else. So that really kind of condensed our season and also just reduced the number of harvests because, we really had to come in and beat those plants pretty hard with the harvester just to get the fruit off that might have been dried up a little bit and shriveled by the heat. We estimate that we had about 30% crop loss or yield reduction due to the heat dome. But we did do 4 harvests between July 24th or June 24th and July 5th. And in terms of yield, again, a little bit lower than usual, we think, because of the heat dome. But our high density planting at 2.5 feet spacing, we got lower yield per plant, but higher yield per acre. So that came out to about 2.9 tons per acre compared to 2.1 tons per acre at the 5 foot spacing, where the individual plants had higher yield, but there's fewer plants per acre. So we had lower yield per acre with the 5 foot spacing. Our primocane suppression didn't affect our yield at all, but that's not surprising because this was the first year that we did it. And there was only a couple months between the primocane suppression and our harvest. However, the berry weights were a little bit different depending on whether or not we did primocane suppression. So the berry weights were average 7.4 grams with no primocane suppression compared to 7.2 grams when we did spray those primocanes. So 0.2 grams is not a huge difference. It may not be very impactful in the long term, but it's something we want to keep an eye out if that's a consistent effect from year over year. Our cull, which is the fruit that we sorted out. So in that bottom middle picture in the cardboard box, you see the fruit that we've sorted out. That averaged about 9% of our total yield. So that again was higher than usual due to the heat dome. We were trying to sort out the fruit as it came along the machine harvester conveyor belt. Might not have sorted out as much as a typical grower or packer. Just, we were doing as best we can, but we're not selling this fruit. So a grower might have been a little bit more particular, but we did try and sort out as much as we could out of there. And our treatments did not impact that at all. The ground losses, so that's the amount of fruit that falls on the ground during machine harvest. We came back after harvest and picked up that fruit and weighed it. And we found that we had less ground loss when we had the 2.5 foot spacing compared to 5 foot spacing. So 15% of the fruit fell on the ground with the 2.5 foot spacing compared to 20% with 5 feet. And that could just be because the catcher plates are staying in a more consistent position with that denser 2.5 foot canopy. Maybe the berries are just kind of bouncing off of the canopy of those plants and onto the catcher plates rather than falling on the ground when there's gaps in the canopy and the catcher plates haven't totally closed around the plant, like we might have seen more with the 5 foot spacing. Now, the top middle picture, you see one of our students was carrying a floricane. So we came through and measured a lot of things like floricane length, lateral number, length of laterals. And you also see Bernadine measuring some of those things on a floricane in the bottom right-hand picture. And we also measured and counted primocanes. So with primocanes, we found that we got more primocanes per plant already with the 5 foot spacing compared to 2.5 foot spacing. So when we grew them at 5 feet, we had 15 primocanes per plant compared to only 9 with the 2.5 foot plant spacing. So obviously the density is already having an effect on the plant growth. When we did primocane suppression, we also found we would get more primocanes when we did suppress them. So 14 versus 10 with no primocane suppression. And that's a pretty typical effect. And we want to continue the study into future years to determine how that might affect yield and overall productivity. The primocane length, they were about 16 to 17 feet no matter what our treatments were. So we haven't affected our primocane length at this point. With our floricanes, we didn't see as many effects of plant spacing or primocane suppression on floricanes. That's not surprising because, last year, which is when those floricanes were growing as primocanes, was the first year after planting. So, we didn't expect to see a huge amount of differences. We did have slightly more floricanes per plant with the 5 foot spacing, just like we saw with primocanes, and expect that to continue. The primocane suppression, again, we didn't expect to see differences in the floricanes because of that, but we might expect to see that in future years. The floricane length and things like number of buds, number of nodes per floricane, there was no differences in those either. We had an average of about 35% bud break, which is a little lower than we might expect. It's usually about 50% in Marion. So we don't know yet if that is just a season effect this year, or if that's something that will just be what we typically find in Columbia Star. And then again, in the number of berries per lateral, we did not see a difference because of any of our treatments, but it was kind of low. We only had about 4 berries per lateral, which is, again, it's less than Marion. Could be a genetic difference with this variety with Columbia Star, or it could have been an effect of the couple of weeks of really heavy smoke that we had in the fall of 2020 from the wildfires, when the whole station was pretty much encompassed in smoke. And that was right during the period of flower bud development. So that could have reduced our number of berries per lateral. And then the last thing I want to talk about is the cane training. So the picture on the top right is our 5 foot spacing August training. So that we kept track of the amount of time it took us to do that training. And when we had 5 foot spacing with the typical August training, floricanes caned out, that whole process took us about 81 hours per acre. Now compare that to doing new over old training, which only took 26 hours per acre at the 5 foot spacing. So a professional crew might take a little bit less time, but we're looking at the relative difference. So that's a really big difference in amount of labor and cost it takes to train those plants. What was interesting was at 2.5 foot spacing, when we did August training, it took 96 hours per acre. So 15 more hours per acre compared to the 5 foot spacing. But when we do new over old, that took the same amount of time as the 5 foot spacing. So no matter the plant spacing, that took 26 hours per acre to do new over old. So that didn't have as much of an impact. So just to wrap it up, with our high density spacing planting at 2.5 feet, we, in the first year of harvest, got higher yield per acre and lower ground losses. But obviously your plant costs are going to be higher, and the training costs, if doing August training, are also higher. The new over old, we estimated it to save, between $800 and $1,000 per acre compared to August training. And when we do primocane suppression, we get more primocanes per plant, and we also have more primocanes per plant at 5-foot spacing. So we're keeping track of all these things, and we want to come up with an economic analysis once we have a few more years of data. So this project is wrapped up. That's why we're doing the final report here, but we will apply for another round of funding from the Northwest Center for Small Fruits to continue this, because we think it's very important to continue these treatments and follow these plants for the next couple of years, to see how things like primocane suppression and training play out in terms of yield and fruit quality and the economics of it, because we really haven't had the time to find that out yet. So we're excited to keep this study going, and hopefully we'll be able to do that with funding from the Northwest Center. And I'm going to kind of minimize my screen there and go back here. So if anybody has any questions, you can go ahead and ask now. We've got plenty of time, I think, to answer those. And so I'll be able to see them if you type them in, and I can answer them. This project is also in collaboration with Dave Bryla. So he's doing a couple of presentations today that hopefully you can catch, looking at blackberry plant water use in Columbia Star. And so his part of the trial is actually right in the middle of this field. So it's important that we keep it going. I know he's applying for funding to keep his part of the project going also. I'm not seeing any questions on my side of things. I'm going to check in with the moderator. I guess if there aren't any questions and people are still here, thank you for coming, and enjoy the rest of the presentations later today. Okay, I got a text from someone that said they don't see how to ask questions on their screen, so that might be why nobody's answering questions or asking questions. If you do have questions for me, feel free to email me. My email is just amanda.davis@oregonstate.edu. So please feel free to reach out, or I think there's a way on the Socio app to message me. Whatever you'd like to do. I'm happy to answer questions in another format that works a little bit better. So thanks everyone for being here.
right. Time to go. Hello, everyone. I'm assuming that this is audible here. Yes. Today, I'd like to present to you a study that I've most recently completed concerning the cultivated strawberry, Fragaria ananassa. We did a phenotypic characterization of a strawberry diversity collection. To advance the slide. Yes, help with advance. I'm sorry. Oh, okay. I was using the mouse, but. Okay, thank you. To describe the diversity of the strawberries that we used in this study, you can see that we have strawberries from 17 countries and 19 states. The main breeding programs that we included in the study was the University of California, Davis, the USDA program in Maryland, and the USDA program in Oregon. This first column shows what we just were mentioning, that in a graphic representation, the majority of the cultivars that we examined were from the United States. The second column shows that the majority of our cultivars were released between 1901 to 2000. The many cultivars that we examined were either thus first or second generation out from the latest patented materials now in commercial supply. The third column shows that mostly we looked at named cultivars. Some of them were breeding lines, and in addition, wild material. Our whole staff of the repository came out to help with the planting of the strawberries. The plants were planted in 12 rows and with guard row plantings around them. There were 3 blocks. The first block in blue was 4 rows. The second block in red were 2 rows on the irrigation wheel side to the left, and 2 rows in addition on the right. And the yellow block was towards the right side further on. Guard rows, as I mentioned, surrounded the sampling. Fruit samples were collected and separated into groups for analysis. Samples were first measured, weighed, photographed, and in the laboratory at the National Clonal Germplasm Repository. That was the fresh fruits were measured. Then the samples were divided into groups to be frozen for sugar and acid analysis, and secondly, for anthocyanin analysis at the Linus Pauling Institute of Oregon State University. The remainder of the fruit was taken and kept for seed extraction. You can see there's a lot of data that was collected, and I just want to show you that there were 15 traits collected for each sample. And of course, there were 3 replicates for each of the cultivar or selection samples. And here are a few summary statistics for the traits. I'm only going to be talking about the fruit berry weight, which the largest berry weight was 45 grams. And the largest total harvest weight for the season was a little over 1 kilogram, 1,029 grams. And again, this was an average of 3 samples. In addition, we looked at runners and we counted the runners put on end to end. One plant produced, the most that one plant produced was 112 meters of runners, and that's an average of 3 samples. And the largest number of runners per one plant was 492. If we look at fruit quality traits, we pulled out cultivars with the lowest and the highest fruit quality traits. To the left, these had some of the lower qualities, and the right circles had the higher ones. In addition, we looked at shortened ripening periods, and these cultivars were chosen for the quickest times to ripen, while the left-hand column or the right-hand column had longer times of ripening. We performed Pearson's correlations with the 15 different data traits and the 2 years of data. Here are 3 examples. In the top, harvest date by bloom date is positively correlated, and the days to ripening compared to the bloom date was negatively correlated there in the middle. And on the bottom, the days to ripen and harvest date was positively correlated. You can see there's an awful lot of data, and I'm not going into specifics here at this point, but we will be publishing this in an upcoming manuscript. And also we have data on GRIN that we're going to be talking about in a little minute too. So the fruit shapes, there seemed to be a trend of breeding over decades to move from globose to conic and conic wedge. The berry weight was not so clearly seen to increase over those decades, but given the cultivars of the study and the climatic conditions, there was higher confidence intervals to make that separation. I would like to mention 3 cultivars that were our quality trait stars. They included Independence, Orris 2427-1, and NW 90054-37. These are all from the Oregon State breeding program. And Independence had the best of both worlds with quality fruit and an abundance of runners, as you can see in the histogram there. I will mention that Orris 2427-1 is a full sib of Mary's Peak. For the ripening stars, the ones that, these are the cultivars that had a quick start and were the ripening stars of the study. They included Oso Grande between 25 and 31 days of ripening over the different years, Cuesta between 28 and 36 days of ripening, and Pinnacle, one of Chad Finn's releases, over 28 to 41 days of ripening. If you look at this runner chart next to it, though, they put their carbohydrates into the fruit. They did not produce many runners for either the 2019 or the 2020 data sets. And to collect our runners, we harvested the runners the week of July 7th. So this was after the plants had finished doing their June bearing, and so where we have cultivars such as Independence, where they did the full fruit production and then had a quantity of runners, that was an interesting point. I would like to mention that this data is present. We have uploaded it to the GRIN, the Germplasm Resources Information Network, and some of you may be unfamiliar with working on GRIN. So I have a slide set here that can show you how to get access to this data. If you go to our NPGS GRIN basic search website, you'll see a front page like this. And there is a descriptor tab at the top. If you click on that descriptor, you can get a list that pops down below, and you can choose the crop of interest. So in this case, if we type in strawberries in that intake box, it will go to the strawberry traits, and you can see that the strawberry is highlighted. It just came up with that. And there's a whole list of descriptors, including the 15 that I took for this study. But if we were to select that box there that talked about total anthocyanins, for example, this is the study that we just finished performing. And if you go to the 2020 data there, you can see that there's 274 accessions that were analyzed. And the data, if you click on that blue, up will pop the different accessions that have that quality. And the value of the data is there on the right where the blue arrow is. And if you click on that value tab, you can sort it. This is the highest sort where the highest values are at the top. Or if you click that blue button, then the lowest values are there. So you can see for the total anthocyanins, the white fruited types had the least anthocyanins there. But you can get this information then through GRIN. And one other very interesting thing, if you check off several trait boxes, you can ask for a particular level. Say you want above 20 grams of weight, any berry that's listed in GRIN that's above 20 grams in weight and above 20 milliliters per 100 gram of fresh weight for anthocyanin, the total amount, we can get a result. And this is the list of accessions that meet both of those criteria. So GRIN can be a useful search tool for anybody, grower, breeder, or people of general plant interest. So in conclusion, we really were appreciative of finances from the Northwest Center to perform this study on 284 different strawberry cultivars. The accomplishments that we made, this data was loaded to GRIN Global. We're going to also be publishing a manuscript on this data. Extra seeds for the cultivars were sent to Fort Collins as backup germplasm. And some of the data, some of the important findings that we had, the largest average runner production per plant was 112 meters of runners for that plant that was the most productive. And the largest harvest per plant per season was, as I mentioned, a little over 1 kilogram under our conditions. Strawberries consistently above the average fruit quality characteristics included Independence, ORIS 2427-1, NW 90054-37. And then the strawberries with the fastest, most consistent harvest were Oso Grande, Cuesta, and Pinnacle. I wish to thank the group of folks that helped with the plantings, collecting data, analyzing data. Dr. Linlin Chang was a visiting scientist from Beijing. She came in 2018 before the COVID situation came, and it was great to have her visit at our repository and help with this project. And Laura Duncan, Brandon Harms, Gabriel Flores, Ryan King, and Todd Anderson of our group helped out with the data collection. Bob Durst from the Linus Pauling Institute at Oregon State University oversaw the anthocyanin analysis, and Zach Weigerd from the Department of Food Science and Technology was very helpful with the food chemistry analysis. And finally, I'd like to acknowledge our funding sources: the Jewish Communal Fund, a USDA ARS Small Fruit Crop Evaluation Grant for 2019, and of course, the Northwest Center for Small Fruits Grant of 2020, and our federal CRIS project as well. So that is my presentation, and I will look for questions at this point. If anyone wants to put some questions in the chat, I'll be glad to monitor.
So you might have to repeat this to the people virtually, but I missed the beginning of this presentation. So is the plans to keep this ongoing to a certain extent, or periodically be adding information to this platform? Can you repeat the question?
Yes, the question was, will this be an ongoing project? And I suppose you're asking whether we'll be adding data to the GRIN system. So firstly, the project. This was a fixed project that we did a 2-year study growing the strawberries out in our field situation. And I might mention that this is only a subset of our total strawberry collection. Doing up to 300 cultivars with 3 reps, 900 was a big effort. And so we've periodically done this kind of thing before. So this was a fixed study, and the data is up there. Also in GRIN are previous studies. For example, we did a similar study to this back in 1985. So that data is there as well. And we intend to do additional studies in the future. And each of the times we do a study like this, the data will be uploaded to GRIN. Also, we are pleased to collaborate with other scientists that are have different conditions. We've uploaded a number of the studies that Chad Finn and Jim Hancock and Jim Luby have done previously on strawberry data. And so in GRIN, these multiple studies are available for searching. Any other questions? I don't see any in the chat at the moment. We also were able to upload a number of thousands of images of the strawberries. The ones we did the fruit images, plant images, and flowering images as well. So we're trying to make a full representation of the characteristics of the strawberry in our GRIN database.
And I may have missed it, but a little birdie had mentioned that you might be retiring in the next little while.
Yes, I'm going to be retiring at the end of March. Thank you very much. I've been assured they'll replace the research leader position at the unit. And we've just recently hired a new curator, and our other staff members, Dr. Nahala Basil, who's our geneticist, and other team members are there ready to take on the cause to the next generation. Well, that's my presentation.
Thank you.
Do you want the slide advance or just the arrow?
I can just use the arrow. Okay, it's fine. So when people are asking questions, will I look? Will I?
So if you'll be able to see the chat here.
Okay.
If you click on anything here,
Yep.
you're going to have to go down here and click on your actual presentation.
Okay.
To
So
regain control.
so don't do that. Literally just use that arrow. And I
If
should
you
be
use
good.
the arrow, you should be good. But if for some reason it's not advancing, you
Yeah.
just have to select it there. And you're good.
Okay.
It looks like it's going, so you should be good.
Okay. So,
Okay, you're good.
okay. So good to go. Alrighty. I'm gonna get the attention of the. Yep. Sounds good. Alrighty. Well, it seems like I've been given the go-ahead to get started. So, all right. I will try to pronounce or enunciate through my mask and into the microphone. Yeah. So thank you for those attending in person. Thank you for those attending virtually. My name is Bill Weldon, and I am currently a postdoc in Dr. Virginia Stockwell's lab here at the USDA in Corvallis. And I started right at the end of 2020, and my primary responsibility has been working on projects around dryberry disease of caneberries. And so I'm here today to talk a little bit about some of the research progress that we made with respect to this pathosystem over the past year or so. This picture that we're looking at here on the right, this, for those of you that aren't familiar with the disease, this is a very classic dryberry symptom. Out in the field, you generally tend to start seeing the disease right around green fruit stage, maybe a little bit beyond that. And what you'll see is the green fruit, instead of kind of that larger, brighter green fruit that you're traditionally maybe used to seeing with blackberry and raspberry, you'll start to see the fruit kind of shriveled. It'll become kind of a matted gray appearance, and this necrosis emerging from the receptacle and up the pedicel just slightly around 5 millimeters or so is really characteristic of the disease. And so there's other, even physiological responses sometimes that you can see in the host that sometimes look like this, but this, the graying of the berry and especially that necrosis seem to be very characteristic for dryberry disease. So since I've been here over the past year, and since Virginia's group has started working with this disease and with this pathosystem over the past few years, one of the first questions that we've tried to get at is a very simple one, which is just what is the causal agent? What is the true causal agent associated with dryberry disease? This disease has been reported in literature and agricultural reports as early as the 1930s out of agricultural reports, especially in British Columbia, Canada. And between the 1930s and late 1950s, there were kind of conflicting reports. There wasn't a consensus on what the causal agent of the disease was. There were some reports that alluded to a bacterial origin. Some suggested that an insect pest might be involved in the pathosystem. And it wasn't until 1959 that a scientist out of British Columbia, McKean, conclusively demonstrated that it was a fungal pathogen that was causing this disease. He was able to reproduce symptomology with this fungus that he classified at the time using the tools that were available to them as scientists of the late 1950s as Rhizoctonia rubi. And this was largely based on just the pathogen's growth morphology on petri dishes, and then its more microscopic growth morphology under the microscope. Since we've started working with this pathosystem in our lab, we've noticed not only that the growth morphology of the isolates that we brought in the field are consistent with the monolinia type fungi, but also because it's flash forward 50, 60 years, we have the advent of some genetic tools that weren't originally available to the scientists when this pathogen was first classified. And so what we're looking at here on the right is just a genetic phylogeny where we took 3 different genes that are commonly used to differentiate fungal species down to the species level. And what we can see here is on the right, all of these isolate names in red are different isolates of the dryberry pathogen that have been brought in from the field into our lab and are maintained in culture. And we can see that not only does it cluster on its own unique branch within all of these surrounding monolinia species, but it seems to be clustering within more of the mummifying type monolinia. So what I mean by that is a monolinia species like Monolinia vaccinii corymbosi, that is the fungus that causes mummyberry of blueberry. And so it kind of shrivels up that blueberry and mummifies the the berry over the winter. That's in contrast to more of your brown rotting monolinia like Monolinia laxa, Monolinia fruticola. And those cause your brown rots on cherry or peach and some of your stone fruits. Apologies. So yeah, it was important for us to figure out. It seems like the causal organism is originally classified as Rhizoctonia rubi, but we're in the process of reclassifying that pathogen within the genus Monolinia and actually naming its own species Monolinia rubi because it does cluster on its own branch within the Monolinia phylogeny quite well. So once, or more or less at the same time, when we were figuring out kind of the true identity of the pathogen, we were also working on figuring out just what are the optimal culturing conditions. Because when we bring pathogens into the lab, we need to, basically figure out what are the media, what's the temperature range, and what are the lighting cycles that are most optimal for culturing this pathogen, so that we can kind of then transition into working with this pathogen in the lab and conducting more hypothesis-driven experiments like the work that you're seeing here on the right, such as fungicide sensitivity screening. But this picture here on the left, just one example of what I wanted to show you is each one of these rows of petri dishes is a different type of culture media, and every column is a different one of the dryberry isolates that we maintain in the lab. And I'm just highlighting the fruit juice based media and your classic potato dextrose agar as types of media that we identified just had kind of optimal rates of growth for the pathogen and are media types that we chose to kind of move forward with for any sort of experimentation that we're going to do down the road with the pathogen. And so the big takeaway there is fruit juice based media, or PDA, somewhere between 20 to 25 degrees Celsius, and the lighting cycle doesn't seem to matter that much. But it was important to kind of establish that base because now we can move forward into more of an applied project like fungicide sensitivity screening, where we're getting at kind of a low-hanging, directly applicable question of, we have these isolates that we brought in from the field. What is their baseline sensitivity to various fungicide groups that are commonly deployed to manage other pathogens within caneberry production? And so the fungicides, all technical grade fungicides that we screened, a few from the DMI groups, so propiconazole, tebuconazole, and myclobutanil. One representative from the SDHIs, we screened boscalid, and then a couple QOIs with pyroclostrobin and azoxystrobin being screened. And these 3 images at the bottom, this kind of highlights this experiment in a nutshell. The leftmost petri dish that you're looking at, that would be a petri dish that is not amended with any fungicides. Those would be our control plates. And each one of these colonies is a different isolate. So we're growing 3 different isolates on the same plate. And what you do is you just make comparisons to petri plates amended with different concentrations of whatever fungicide you're interested in screening for sensitivity. And so we ranged everywhere from like 0.01 to 100 micrograms per mil, microliter, micrograms per mil. And the big takeaway there is that our isolates that we have currently in culture, which are kind of a representative, baseline sensitivity of the population as far as we understand it, are that they are sensitive to this entire range of fungicides at concentrations well below field rates. And this hints to potentially why this disease has been able to hang around at relatively low levels since its kind of initial reports in the 1930s, as some of these conventional fungicides are typically applied to control other diseases such as gray mold and rust. And they're kind of potentially having a secondhand controlling effect on dryberry disease that's keeping it around at low levels. Although you do see, certain situations when that perfect storm comes along, maybe some organic production systems that aren't able to use some of these synthetic fungicides, or just a new planting where they're light on the chemical control, you can still see this disease take off. Another kind of initial question that we wanted to get at is just when is the spores of this pathogen, when are they produced throughout the growing season? Maybe get a better sense of where the season might be, and that if there's a bunch of this pathogen spores in the air at a time when it's berry tissue is available, and that would be a very optimal infection. And so we were interested in just an entire shed over the course of the entire growing season of when Monticello rubi might be in the air. And the way that we went is we deployed these rotated spore samplers, and you can see this looping video as just an example of how this looks out in the field. But these are PVC sampling arms deployed in collaboration with Lisagen, and on that little on the bottom there are two stainless that are greased with just like a plumber's grease, and as they spin, any of the spores that are floating around in the air, small insects that come in contact with the rods, will be impacted and they'll stick. And then when we switch that out, we have a snapshot of the population that was floating around in the air at that time. And the start of the growing season, and did this all the way up to harvest, and these 3-day intervals, therefore kind of giving us a snapshot of the spore population throughout the entire growing season. And so right now, the DNA extraction, so we take those rods that have spores, we bring them back to the lab, we do DNA extractions, similar to how a PCR nowadays, you tip up your nose, DNA extract, and then they're looking specifically COVID virus. We're bringing our spore rod back into the lab, doing a DNA extraction, and looking specifically for Monticello rubi using a molecular-based PCR, very similar to the COVID test. And so that is underway. We've had to do a little bit of optimization of, say, the assay was only able to detect down to the level of the entire Monticello genus, which would include spores theoretically from Monticello vaccinii corymbosi, or the brown rot, the cherry brown rot, or the peach brown rot, the Monticello laxa and fulla. And so I've been working recently on optimizing a PCR assay that's very specific to just Monticello rubi. So the PCRs back in the lab, we know exactly which Monticello we're getting. And so it's something that's ongoing, and that's something we're going to continue to, subsequent growing seasons, so that we'll get multiple years that we, in theory, our hope is correspond specific times of year or weather conditions for what might be in the air, help guides associated with those peak periods of risk, essentially. And, to take any questions from anybody in person or in the audience. Other ongoing projects that we have, our lab is currently getting the entire genome isolates sequenced, utilizing PacBio and Illumina-based sequencing. That project is ongoing. It's been a little bit slowed down due to COVID-based shutdowns with sequencing facilities, but it is ongoing, and it's making good progress. And So yeah, with that, I'm happy to take any questions that anybody may have, and I thank you all for your attention.
So I apologize if you mentioned this and I missed it.
Yep.
So you kind of mentioned the disease has been in and out at low levels for a
Mhm.
long time. Was there a recent uptick or like regional variation that's kind of been the case for now?
Yeah. So the question, the question from the audience is that we're touching upon how I said that it's, it was noticed in the early 1930s and it's been hanging around for a while. And basically, what's the reason for the renewed research interest? And it's primary. There hasn't been like any sort of massive epidemic that rolled in 3 or 4 years ago that like kind of renewed that interest. It's a disease that has been hanging around. Fungicide-based resistance is not an uncommon thing for some of these pathosystems, and it's just interesting that a monolinea type disease, which was not previously reported in caneberries production or on caneberries, now seems to be documented, and other monolinea diseases, can be pretty, have pretty impactful reductions in yield. And so, it's in the best interest of this region, which is where all of kind of the incidents and severe, reports of this disease historically have been, to just get a better understanding of kind of those baseline ecological and disease management type questions, so that if it were to start to reemerge, we at least have an initial game plan for the disease. Cool. If anybody has any questions in the remote audience, I'm happy. I have the chat screen in front of me, and I'm happy to answer any of those questions as well.
I was going to say that with this disease, it was just something that kind of stood out. Those who, just standing in caneberry field for hours.
Yeah.
Looking at stuff, because we were doing a microbiome part, but it was like, what is this?
Yeah.
What's going on? Why are these berries dying? Some of the laterals you'd see 30% of the berries filled. So it's patchy, you feel. And then we started doing the molecular characterization, and it was not hitting anything except kind of a monolinea. And then there's a serendipity. Eventually, when I just reran the sequence again, all of a sudden it's 100% match to Rhizoctonia rubra.
Okay. Thanks.
And just left and right for the slides to advance.
Yeah, and this
Or that guy
working
there
too.
too. Yep. You just got to make sure that you are flipping. Actually, you're on the slideshow, so you should be good to go.
Okay. So I'll get going here pretty soon. Some people might filter in, but it's a small group. I'll keep it kind of informal.
Hey, is this one going to be better than the one this morning?
I know you were here this morning. There's no new information, but yeah, I'm happy to be here to present work that really was largely done by Dr. Rachel Lyso and Bridget Jarrett. Rachel was a professor at the Montana State University Western Ag Research Center with me, and Bridget Jarrett has been our berry program manager and lab manager for a long time. Yeah, so my name's Zach Miller. I'm the director of and professor at Montana State University for our Horticultural Research Center that's over in the Bitterroot Valley, south of Missoula. And we are the one horticultural research farm in the state. So we have a really diverse portfolio of research that we do for our mostly small scale producers. And I came over there in 2014 after doing a postdoc on the main campus to really revive the center. There were no faculty there. At the time, they'd all retired. It's actually one of the oldest ag research centers in the state. It was started in 1907 to support the major apple boom that was going on at the time in the Bitterroot Valley and some other of the valleys of Montana. It's amazing to see the old pictures from that time. In our one county, there was 15,000 acres of apple orchards in the 1910s and 20s. And there still exists a small apple industry there. Anyways, I came over. We've established a program in perennial fruit horticulture, really diverse. One of the big projects that I started initially was looking at some of these sort of novel cold hardy fruit types. So we were looking at ways that we could capture kind of the growth of the blueberry market, because everyone's looking for superfood fruits, high antioxidant fruits. So we planted Haskaps or honeyberries, Saskatoons or serviceberries, currants, aronia, elderberry, and a few other things, really to look at their cold tolerance, because we in Montana, we're pretty much all zone 5 is the warmest we get. There's a few zone 6 and zone 7 right at the corner of the state. But really we're trying to look at crops for zone 5 or below. And also, berry crops that can tolerate neutral to alkaline soils. And in a lot of these novel fruit crops that, they get introduced, but there's not a lot of real hardcore production horticulture. Like, what will they produce? What are the pest problems? How easy are they to harvest? How long will they store? And really, we're trying to fill that gap for our producers and then share that information across the state. So we've been doing this with all these different crops. They've been in the ground 7-8 years now. We're getting to peak yields. We're really learning a lot about the different crops, and we've done this work with Hascap because it's been one of the favorites. Hascap or honeyberry is an edible honeysuckle. Who here has tried one before? You've tried some in Bozeman?
And Minnesota.
Yeah. They're they've been grown for centuries in Siberia and Japan, are native to around the northern hemisphere in cold climates. So there are wild Lonicera caerulea is the species name that grows in the Cascades, in the high mountains in Idaho and Montana, and then also up into the boreal forest in Canada. So yeah, it's been and it's super cold hardy. It's like zone 2 hardy. It'll take 40, 50 below. The blossoms are come out shockingly early for us, so it'll start blooming in March or April. The blossoms, even when it's in full bloom, it can take mid-teens and still produce fruit. It's amazing to see the bumblebees flying around, and it's mainly bumblebees that we see pollinating these. They're flying around in 40 degrees, 50 degrees. They're out there working them, and we get great fruit sets. So we've done now a lot. In the past, we did a lot of work on just productivity, which yields the best, worked with food scientists and with consumers to do taste tests. These have been, and they've been quite productive, at least in our environments. In many of the mid to higher yielding cultivars, we're getting 4 to 7 tons an acre, which is fairly good for a berry crop. Yeah, like I said, they've been doing these basic variety evaluations are important because they are really diverse. So this is just an example of a few of the Hascap or honeyberries that we have planted at the station. We have many of these Taka, Tana varieties that came from Maxine Thompson's breeding program here in Corvallis area, with Sinji Kawai, which this one's named after. Cheeto is one of hers. A lot of her varieties, also some of the varieties that have been produced in the University of Saskatchewan and Bob Bohr's program. And then some of these like Blue Goose, Sugar Mountain Blue, and Wild Treasure, which are more Russian origin. And especially with these novel crops, there's a lot of varieties that have been released without much screening. And a lot of the early varieties that were released in North America in the 90s were not the best representatives of what this fruit can be. A lot of these Russian types are very small, very, very early, and often have odd medicinal flavors. But Maxine and Bob have combined a lot of the Russian and Japanese gene pools to produce larger fruit that are much more commercially viable, much more productive, much better flavor. As we've gone through these, looking at these for their commercial potential, Hascaps out of all those cold hardy berries were by and away the favorite because they have they're a great fresh berry. So they've got a much larger potential for the fresh or frozen market. Also a lot of processing. It's healthy candy, great tasting, and so for this project, we went beyond just that basic horticulture of which tastes the best, which produce the best, to look at some of the issues with how long can you store these? I mean, you're going to get your top dollar for a fresh market. A lot of the growers we're working with are selling direct, small scale farms. How long can you keep these things in a walk-in cooler? So high humidity, fairly cold cooler. How long can you keep them and still sell them? What's making them go bad? And then we also looked at some potential for mechanical harvest by looking at which berries hold up better to mechanical harvest and simulated mechanical harvest. So that's what we did for the research that was funded by the center. If you have any questions, just jump in and I'll try to remember. So initially, the first trial we looked at was, so these novel crops, there's really not much known about their ripening physiology. Rachel had done some preliminary work looking at, do they produce ethylene? Could they maybe ripen in storage? And we saw some ethylene in that. So we looked at harvesting them really unripe, where they're just starting to get color, or when we typically harvest them when the berries are starting to soften a little bit, when you're getting to mid-teens for Brix. So we did this with 2 different varieties. And really, those unripe berries stay unripe. That is the take-home message. But there is some silver linings to this. So yeah, those berries that were half blue were much smaller. And while they did ripen, like the Brix went up, the acidity went down, but they didn't ripen to a point where you'd want to eat them. But the silver lining part is that in varieties like Tana, which is one of Maxine Thompson's cultivars, when you pick it on the bush, it's a little sour. It's more closer to a raspberry than a blueberry. But with 2 weeks in storage, that acidity has dropped down quite a bit. And this is a better tasting berry after sitting in the cooler for a week or 2 than it did going in or fresh from the field. We didn't see as much of a drop. We still saw a fairly substantial drop in Aurora, which is closer to a better balance of acidity to sweetness in the field. But yeah, for a lot of these varieties that are a little bit more sweet tart, they do get better with storage. But can you keep a berry for 2 weeks without it going bad in the cooler? The answer to that was yes. So this is the percent of berries that had any sort of mold, botrytis, spoilage on them at 2 weeks and 4 weeks. We did this over 2 years. The first year we just did it over 2 weeks, and there was really almost no spoilage over 2 weeks. So in the next year we did it over a full month. And really, there are some variety differences here, but any of the varieties you can store in the cooler for 2 weeks, no problem, with very little spoilage. And some varieties we had no spoilage after a month, which is crazy for a berry. And it's kind of it may be related to the unique type of fruit that Haskaps are. So it's a honeysuckle fruit. So you have the twin flowers, like in a normal honeysuckle, that makes twin berries that are then surrounded by another layer of skin that develops from the base of the fruit. So you have this many layers of skin. It's also why they have really high phenolic compound concentrations. They have antioxidant potentials that it's 3 times blueberries, because you have most of those compounds are in the skin, and you have many layers of skin, 3 layers of skin going through that whole fruit. And all of those, I mean, the plant's not making those compounds for us. It's making it to, a lot of those are antifungal compounds. It's making it to attract birds, which definitely come. But yeah, I mean, we think that that's in part why we get such long storage. We also, so while molds were not the thing that's going to ruin them in storage. What it was, for the most part, was just desiccation. We would get, and so that's what this table shows is, and really this is the important part. This is the time to where 10% of those berries in a half pint were starting to shrivel a little bit. So that's probably about as long as you'd want to keep them on the shelf. But still, we've got, 10 days to 2 weeks, depending on the variety for that shriveling. But this is really, it was desiccation that led to the spoiling of those fresh fruits before any of the fungal pathogens were ruining them. I think that this is a useful result for folks who might want to grow them. If you're going to do gentle hand harvest, they will store a long time. There are some variety differences, but in general, it's easy in this crop to get 10 to 14 days of storage life, which is a good amount for a fresh berry. Again, this is in Montana climates where it doesn't really rain. I don't know if you get the same growing it here, but it really adds to the picture of which ones of these could be commercially harvested. So with Hascaps, you make your own market because most people have never tasted them. They don't, because they have so many layers of skin, they don't dry as well. We've played around with dehydrators. It's easier to make like a fruit leather or something like that. They're a little bit harder to dry into a raisin. But they do taste great. Yeah, so we've looked at that. It depends on the variety. So varieties like Aurora have a pretty dry scar, and there's a whole that's a whole wide open area of research because it also depends on when you're harvesting. We don't really know what the optimal harvest time is for these. We've just always gone. I mean, we're sort of making it up, right? Because there's breeding programs that are producing these new varieties, and not many programs are looking at just how do you manage them in the field. But some do have a really wet scar. Some others, we just had a paper in Hort Technology come out. Some of the varieties have pretty large problems with pre-harvest fruit drop. So there's some varieties, not many, but there are some varieties that will drop half their fruit before you even get in to pick them. They still produce a lot. But I mean, that's the kind of basic questions that we're answering. So now for less gentle harvest. So that was all fresh market. How long can you keep them? How long can you maximize that value for the fresh market? And it seems like a long time. We wanted to look at which ones have a better potential for machine harvest, for more of a processed market. And we do not have a very fancy harvester, and we're working more with small scale growers. So we do a lot of sort of DIY farm hacky stuff for folks that are growing on, an acre, maybe 2 acres. What could you do when you're not going to buy $100,000 berry picker? So this is the system we use, which is a cordless reciprocating saw with a little tool adapter, and we make these padded forks, and we just shake the trunks. On our website, we've got Rachel did a lot of great work on just the labor involved, and you can harvest this way pretty quickly. For small scale producers, they've been really excited about it. You can harvest a plant in less than a minute and get 90% of the fruit in some of the easier to harvest varieties. So you can harvest it into these trays or into just a kiddie pool that you cut in half. This is $2,500, and this is less than $20. For some of our growers, they go the kiddie pool way, which actually works better. I mean, these things are nice and fancy, but if it's a hot day, metal trays get too hot and you start cooking the berries and it gets sticky and gross, and a little baby wading pool stays a lot nicer. It doesn't heat up as much. Anyways, for this, what we looked at was the damage that's happening in this process, just shaking them off the bush, and then separately what's happening in this process where we're cleaning all the leaves and debris out of it, just to kind of get an idea of which varieties come through that better, and also which steps of the process they're more prone to damage. And so that's what this table shows. Is this is hand harvesting for 3 varieties. Hand harvesting alone, and the damage was minimal and mainly bruising and cracking in the berries. Just getting shaken off the bush with the reciprocating saw, and then that with that blower, with a leaf blower, removing the debris. So the main take homes here are that the damage is quite low in just that the first steps. Often it's not different, or there's not a lot of increase in damage. There is for some varieties, but not others. Indigo Gem is a smaller, rounder berry, and it's more resilient here. But most of the damage was coming in this last step of cleaning. But in larger berry varieties like Aurora, that damage is quite a bit higher. But again, this is just for a flash frozen processing market. Probably doesn't matter, but there are variety differences. We also looked at a whole suite of varieties through another simulated mechanical harvest, where we were dropping the fruit from a height and then rolling it through a simulated processing harvester contraption. Again, there we didn't. We saw differences among varieties. They weren't necessarily consistent between seasons, but we did see certain varieties were consistently less damaged. Were less damaged, and for the most part, it was the obvious thing. It was smaller, berried, rounder varieties that went through that better. But in this crop, there is such it's new, and there's a wide diversity of genetic resources that could be put into this to optimize varieties for whatever market. And we're just using the first few real the first varieties that have some commercial potential. We're just screening those, and already there's quite a bit of difference there. And with that, I'll take any other questions. If you've the other interesting anecdote is that about cold hardiness. We had it's a lot colder in Montana, and most of our cold injury is fall and spring cold injury. It's usually not midwinter cold injury. We've not seen any cold injury in our berry and small fruit crops in the last 7 years, but last year we had a very mild fall, and then in mid-October it went to 10 below, and almost all of our cold hardy grapes were completely, all the fruiting buds had died. The plants themselves are okay, but they're being regrown. Many of our apples died back to the ground. Raspberries, we grow a lot of cold hardy raspberries like Boyne. All those floricanes were completely zapped. But the honeyberries and all these other cold hardy berries were fine. We didn't really see any dip in production. No visible cold injury. They took the worst that could get thrown at them with below zero temperatures in November, when they weren't quite dormant. The other great, one of the other advantages or reasons people are interested in honeyberries are that they are much earlier than blueberries. So they we'll harvest, and there's a great range in when they mature. So we'll start picking honeyberries in late June, early July with the earlier varieties, and then have different varieties to pick until mid-August. And so it's a berry crop that especially in warmer climates that could be well ahead of blueberries. Drawing in pollinators and providing additional fresh market early in the season.
So with your later cultivars, have you seen any incident with disease severity or anything like that, or an uptick in disease incidents?
So,
Warmer temperatures?
yeah, we're in, a lot of people like the edible honeysuckle Hascap because it doesn't really have a lot of disease and insect pest pressure. We have not seen, had to spray for no pests, and I keep these crops fairly tightly managed because I don't want it to be a train wreck, and I want to figure out what works for people who want to grow them. And I haven't had to spray a single thing on honeyberries. My colleagues in the Midwest who grow these in the Dakotas and in Minnesota do have some problems with spotted wing in the later cultivars, but we consistently trap. Well, we always monitor for spotted wing, and we consistently trap a few individuals in late August, early September, and then it freezes and they go away. But I've seen no fungal disease, no bacterial disease in any of these. It's the real pest management headaches for these are weeds and birds. In the other crops, it's really the ones that are where we have a lot of wild populations or wild relatives that are closer by. So like in any of the ribes, we get cane borer, we'll get gooseberry maggots. In the service berries, the Saskatoons, they're a bear. They've got a couple pests that are pretty hard to manage that are always there. But these are pretty easy for us to manage. But again, we're cool and real dry. It's less than a foot of precip all year. But yeah, it's an interesting crop because you are creating your own market, because most people have never tried it before, but they are delicious. And they are, they have some commercial potential, and we're just trying to hack out what that might be.
You're growing like a lot of the different berries, like a lot of the different sort of specialty berries as like, I don't know, to try out.
Yeah, so we have about 50 different varieties in those 6 or 7 different cold hardy berries and small fruits that I talked about at the beginning. And we've added more Hascap varieties as new ones have come out. They're not in the trial, but we're keeping track of them. And yeah, so many of those. We also do, we've got cold hardy wine grapes and a bunch of different cider apple cultivars. But
Is this one of the more promising, I guess, crops that you're trying?
yeah, the growers have been more interested in honeyberries because there's still the fresh market potential. So currants kind of taste gross when they're fresh. Some people like them, but for the most part, it's a processing berry, right? Service berry, the commercial cultivars are delicious. They're closer to a blueberry. I mean, the wild ones taste like sweet sawdust. But the commercial ones that are basically wild selections from prairie provinces are much more palatable. People really like them, but they're still a little plummy. So it's like a smaller market yet, and they store terribly because they always have a wet scar. And they go bad within a week. But yeah, so growers are more interested in this because you still have the fresh market. You can capture that value at a smaller scale, and the processing for these and the marketing you can do based on the potential health benefits is always there. And yeah, and so we definitely acknowledge the center and all the breeders in Saskatchewan and over here that have helped bring this closer to a commercial new crop for colder environments. Let me check online to make sure. Chat. Nope, no chats.
All right. Hi, everyone. Good afternoon. Thanks for coming to my presentation today. I'm going to go ahead and share this full screen. So I'm going to go ahead and give my talk. And if you have any questions in the meantime, just go ahead and type them in the chat. And I'll also have my email address at the end on the last slide, so you can email me if you have any further questions after this. So I'm presenting for myself and Bernadine Strick on our project in alternative management of Columbia Star. So we're looking at planting density and primocane management and how these things affect yield and labor costs. The objectives of this trial are, again, to look at yield, fruit quality, and labor requirements when we do things like changing the plant spacing, doing primocane suppression, and looking at different pruning and training methods. And this is also a collaboration with Dr. Dave Bryla at the USDA, who you might have heard give his presentation on looking at plant water use and deficit irrigation in Columbia Star as well. This trial was established originally in the fall of 2019. We had kind of a hard frost right after planting, so we did do some replanting in spring 2020 for the plants that didn't quite make it. And it's being drip irrigated, fertigated, and then we're maintaining just bare soil between the rows, mostly with herbicides, a little bit of tillage right at the beginning when the planting first went in. So you can see kind of from planting to the first fall of 2020, those were the plants surrounded by all the smoke that we had from the wildfires last year. Here are our treatments. The first one is plant spacing. So 5 foot in the background there, that's kind of what we're considering the industry standard. And comparing that to the 2.5 foot spacing in the front there. So twice as many plants per acre. And that's just the summer of 2020, not long after the replants. By spring of 2021, these plants grew really well in 2020. We were happy with that. And they kind of bounced back from any inconsistency in size that we might have had from the different planting times. They were all pretty uniform by the fall of 2020. And in spring of 2021, we see a pretty solid canopy there with the 2.5 foot spacing on the bottom picture compared to a little bit more gaps in the canopy and just less uniformity of canopy in the 5 foot spacing as of spring 2021. Another of our treatments was primocane suppression. So the picture on the left is without primocane suppression, and the one on the right obviously is with the suppression. This was one application of AIM a couple weeks before this photo was taken. And the goal is to try and increase machine harvest efficiency by having some of those lower fruiting laterals out of the way. And this can also, in some cultivars and past research, increase primocane number per plant. So we wanted to know what would happen in Columbia Star if we tried primocane suppression. And finally, training method. So we're doing either the typical August training where we cut the fluorocanes out after they've started dying off after harvest, and then training the primocanes up on the trellis in August, or doing the new over old system that many growers have started using to try and save money and labor costs, where we just leave all the fluorocanes on the trellis for several years in a row and throw the primocanes up and over the existing foliage plant material that's on the trellis. So this does save quite a bit of time, which I'll talk about at the end of the presentation. But it is concerning in terms of disease. We know that the leaves and the canes can harbor a lot of disease, fungal issues, fungal pathogens, especially with the concern around the blackberry collapse disorder or disease, where this is something we're going to be keeping an eye on. So 2021 was the first year we did primocane suppression, the first harvest year, and we started those cane training treatments that I just showed you the pictures of. And then we collected data on things like cane biomass, fluorocane length, primocane length, and then how long each of these different aspects of management cost. So this was a picture, June 24th was our first harvest. The fruit was looking really good. We were lined up to have a pretty good yield for our first kind of baby crop season. And then along came the heat dome. You all know it. It was obviously really terrible for the industry. And we felt the effects of that too. This was a screenshot from Bernadine's phone. We were in the middle of harvest at 7 a.m. and it was already 84 degrees. We know it got up in the, well above 100 degrees and really did a lot of damage. So this was July 3rd, a couple days afterwards, looking at our research field and the dried up sunburned fruit, the damage to the leaves that we saw there. So our harvest was a little bit compressed. We only did 4 harvests between late June and early July. And we are guessing that our yield was about 30% lower than usual because of that, through smaller fruit, shriveled fruit, and fruit that just wouldn't come off of the plants. We had to turn the beaters on the machine harvest up pretty high to get some of that fruit off, just because it was really sticking on the plant more than usual. But we did see some treatment effects even in this first year. So when we had plants growing at 2.5 foot plant spacing, we got a little bit lower yield per plant, but higher yield per acre because there's twice as many plants per acre. So that one had 2.9 tons per acre when we extrapolate that out to a larger plot size, compared to the 5 foot spacing where we had almost 5 pounds per plant, but that only came out to 2.1 tons per acre. So some pretty big differences in yield per acre in the first year already. Our berry weight was not affected by plant spacing, but it was slightly higher when we did not do primocane suppression. So it was about 0.2 grams larger without primocane suppression compared to the plants that we did do the primocane suppression. It's not a huge difference, but it's something we want to keep an eye on because it can make an impact, as yield increases, and just that might be something that becomes a larger difference over time also. Our coal, which is the fruit that we sort out on the machine harvester itself, and you can see that in the cardboard box that's in that crate. That's the fruit that's getting sorted out just as it's coming down the conveyor belt on the harvester. It's about 9% higher than usual due to the heat dome. We tried to sort out as much of the really badly damaged fruit as we could while still trying to get some good yield data from the first year of harvest from this trial. Our ground losses were a little bit higher with the 5 foot spacing, so 20% of the fruit falling on the ground after harvest compared to 15%. And this is likely because of the catcher plates on the machine harvester closing pretty tightly around that solid canopy in the 2.5 foot plant spacing. And it's possible that some of the fruit were just bouncing off that canopy and going onto the catcher plates rather than falling through the cracks in the catcher plates as they moved around those 5 foot spaced plants. For primocane growth, when we had wider plant spacing, we got more primocanes per plant. So 15 per plant when at 5 foot spacing compared to 9 per plant at 2.5 foot spacing. This was really evident, especially when we were training these plants, which I'll talk about later. However, our plant spacing and our primocane suppression did not affect the length of the primocanes. So these were all about 16 or 17 feet, definitely shorter than a cultivar like Marion, and might have been reduced a little bit by the heat dome and just the stress that put on the plants. So we'll continue to measure that too and see what might be more common for length in a typical year, assuming we have a typical year in the next couple of years and the study can continue. Our primocane suppression also resulted in more primocanes per plant, which is sort of what we expected, but we didn't know for sure that that was going to happen in Columbia Star. So a few more primocanes per plant when we do primocane suppression, and again, when we have 5 foot plant spacing compared to the higher density. Looking at fluorocanes, we really didn't have a lot of treatment effects on the things that we measured on fluorocanes, except the number per plant. So just like with primocanes, we had more fluorocanes per plant at 5 foot plant spacing compared to 2.5. However, the length of the fluorocanes, the number of nodes, and number of laterals per fluorocane, those were all pretty much the same, no matter the plant spacing or the primocane suppression. This isn't surprising, especially with primocane suppression, because the first application had just gone out a couple months before harvest. So there wasn't really time for that to affect the fluorocanes, which grew in 2020. We did have lower biomass per plant at the high density spacing. But again, that's expected. We have fewer fluorocanes, so it would be expected that the biomass is also lower. Our bud break was pretty consistent, around 35%. That's lower than something like Marion that typically has a 50% bud break. But again, this could be something that just happened this year. The plants are young, or it could be a genetic difference. So that's something we want to continue to look at also. And then the number of berries per lateral was about 4 all across the board, which is again lower than a cultivar like Marion, but with consistent. So we think that this might have been partly attributable to the smoke in 2020, which you saw in that picture earlier. We had a heavy, dense layer of smoke at the research station for a couple weeks in fall of 2020 because of the wildfire smoke. And that was during the period of floral bud development. So that could have definitely impacted our yield and our number of berries per lateral. For the cane training, looking at our labor, this was us measuring the time it took for our crew to do the training. So we recognize that any kind of commercial professional crew may do this faster, but we're looking at the relative time it took for our folks to do this training. So when we have 5-foot spacing and doing August training, that took 81 hours per acre. Compare that to doing the new over old training, that only took 26 hours per acre. So a huge time saving at the 5-foot spacing. Now let's add in the 2.5-foot plant spacing. When we do August training, that took 96 hours per acre. So there's fewer canes per plant, but those plants being so close together, it's definitely challenging to tease out the primocanes from the neighboring plants without damaging them. I helped do this too, and it was definitely harder than doing the 5-foot spacing. So that's reflected in the additional time it takes per acre because of the high density spacing. But it was really interesting because when we do new over old, there's no difference at all in terms of the number of hours per acre. Again, 26 hours per acre, and a huge time saving compared to August training with that high density spacing. So to wrap that up, we had higher yield per acre and lower ground losses when we planted at high density. But obviously you're going to have higher plant costs and higher training costs if you're doing August training. So you can see the yield difference and the estimated cost, additional cost for August training with high density. When we do new over old, that saved an estimated $800 to $1,000 per acre compared to August training, depending on the plant spacing. And we got more primocanes per plant at 5-foot spacing and when we did primocane suppression. So that's something that can and likely would impact yield in the future. And we want to continue looking at that. And then the bottom, we're just keeping track of how much the primocane suppression will cost too. And we'll add that into an economic analysis when we get done with this trial. So what's next? This is our final report for this round of funding from the Northwest Center for Small Fruits, which is, we're thankful for their funding for this research. However, we only have 1 year of harvest. So we really need to continue this to determine what effect those primocane suppression and training methods have on yield and fruit quality. And additionally, what difference the plant spacing has on those things over time also. We only have the first year of data. So that might change yield and the plant growth as the plants mature and they start competing with each other. And ultimately coming up with the most economical way to produce Columbia Star and giving the growers some really good options and background information on why we're recommending the things that we will recommend. So we are applying for another round of funding from the Northwest Center to hopefully continue this work and continue collaborating with Dr. Bryla on his irrigation projects as well. So thank you for listening, and my email address is there if you have any questions that you're not able to ask through here. But I'm going to go back to the Socio platform and see if any questions came in. I'm not seeing any right now, so feel free to type any questions that you have in, and I will be here to answer them. And if there are no questions, again, thanks for listening, and look forward to seeing you all in person, hopefully next year.
Cool. Alrighty, well, it's 4 o'clock and this is a presentation spot, so I'm gonna get started. I think everything is working. Cool. Alrighty. Well, my name is Bill Weldon, and I am currently a postdoc in Dr. Virginia Stockwell's lab here for the USDA in Corvallis. And I started in Virginia's lab a little over a year ago, right at the end of 2020 in November. And one of my primary research kind of focuses that I've been working on while I've been in the lab so far is various aspects of dry berry disease of caneberries. And so I just want to run through some research progress that we've had in the lab over the past year, and then I'm happy to take any questions or share any ideas or anything like that at the end. This picture here on the right is a pretty characteristic example of what dry berry disease looks like out in the field. It can get misconstrued with some physiological responses by raspberry and blackberry as well. But what it looks like out in the field is you typically don't see any symptomology until about the green fruit stage. And then instead of maturing into the pink and red or black raspberries, it will start to mummify, similar to if you're familiar with mummyberry disease of blueberry. It kind of hardens up, it turns a lighter gray, kind of like a leathery texture. And then this necrosis emerging out from the receptacle of the berry and up into the pedicel is very characteristic as well. So those are all things to look at. Obviously, this berry is not viable. And so any berries that are symptomatic with dry berry disease directly result in a yield loss. And so, yeah. When Virginia started working with this pathogen, the first kind of most important question that we wanted to focus on was just to clarify what is the causal organism underlying this disease. This disease has been reported in northern Washington and British Columbia and other parts of Canada as far back as the 1930s. In those reports, anywhere between 30 to 50% yield reductions in yield for that growing season have been reported. And between 1930 and the late 1950s, the causal organism associated with this disease was kind of up for debate. There were some reports that attributed it to a bacterial pathogen, others associated it with an insect pest. And it wasn't until 1959 that a scientist out of Canada, McKean, kind of conclusively demonstrated with Koch's postulates that it was a fungal pathogen that was causing this disease. He was able to extract the fungus, isolate it, and then repeat the exact same symptomology. And at the time, using the scientific tools that were available to him, which was largely just its growth morphology on petri dishes in culture, and then also more of its microscopic colony morphology, he made the designation and classified the organism as Rhizoctonia rubi. And that's how it stood up until our group in the Stockwell lab started looking at it again. And when we started pulling isolates from the field, bringing them into culture, and now flash forward 60 years or so, and there's the advent of some molecular sequencing technology that's greatly improved since that time. It's become really evident that this is not a Rhizoctonia pathogen. This is actually a fungus that falls into the genus Monolinia. And so that is more closely related to other Monolinia organisms. They kind of break up into two clades, you could call them. There's the mummifying Monolinia species, and those, the one that people are most familiar with is Monolinia vaccinii corymbosi. That's what causes mummyberry of blueberry. And you get kind of that drying, that shrinking, that mummifying symptomology with those species. And then there's also the brown rotting Monolinia. So that's your Monolinia laxa and your Fruticola. Those are what cause brown rots on cherries and peaches. And you get kind of that just like wetter brown rot of the fruit. And our Monolinia species, we're designating as Monolinia rubi, because all these isolates that you see in red, these are isolates that we brought back into the field have maintained in culture, sequenced a few different gene regions that are commonly used to designate fungi into their species groups. And they clearly fall into their own group within the mummifying Monolinia part of the Monolinia genus. And so we're going through the process right now of reclassifying this organism as Monolinia rubi, which has, from a scientific perspective, some implications just on how we expect the pathogen to behave out in the field. And so that's kind of an important reclassification to make initially when studying this disease. I think I need to click on PowerPoint. Okay, cool. So after, or more or less at the same time as we were kind of working on clarifying what is the actual underlying causal organism, we had brought isolates into the lab, and this is the first time that anybody's really done research on this organism in about 50 or 60 years. And so we just needed to redo some of those baseline, just define what is the best culturing practices for this organism so that when we start conducting more hypothesis-driven experiments, we know what growth media is the best, what temperature conditions we should be growing this pathogen at, what are the lighting conditions that we should be working with. And so I did all of that both in solid-based culture and also liquid broth culture. And the one kind of picture that I wanted to highlight here, if you look at this infographic, every different column is a different isolate of our Monolinia rubi pathogen, and every different row is a different culture media. And so visually, you can just kind of see that some of the plates have larger colonies and faster rates of growth on those. And so the one we've moved forward and working with are fruit juice based media, like tart cherry juice and blackberry juice, which makes sense because this is derived from cane fruit. And then also just your classic like potato dextrose agar media. And so kind of had our baseline of what we should be working with this in the lab. And now we can move on to kind of some more pressing questions. The first of which was, what is the baseline sensitivity of this pathogen to a range of fungicides that are commonly used and applied to caneberries and cane fruit throughout the region? And so we screened the sensitivity of over a dozen isolates for their sensitivity to fungicide chemistries from the DNMI group, so propiconazole, tebuconazole, myclobutanil, one representative SDHI so far. We've screened it against boscalid, and then a couple QOIs, pyraclostrobin and azoxystrobin. And in the lab, what this looks like is we're comparing non-amended Petri dish plates, some media with no fungicide added to them. This Petri dish here has 3 different isolates of Monolinia rubi growing on it. And these are the same isolates on fungicide amended plates. And you can see that their colony diameter is reduced because that fungicide is effectively reducing the ability of the pathogen to grow. And so we're just looking for the point where that growth is reduced by 50%. And we kind of call that our effective concentration at 50%. That's just a common cutoff that we use in the lab. And when we looked for those EC50 values, the big takeaway here is that all of our isolates that we've brought into culture over the past few years from Northern Washington, those are sensitive to each of these fungicides at concentrations well below the field rates. And so that kind of, at least at a baseline level, tells us that the fungicides being used to spray primarily for other diseases like gray mold and rust seem to be having an effect, kind of a secondary effect on just keeping this disease at bay. That's kind of our working hypothesis, that the fungicides being sprayed for these other more economically important diseases, at least currently, seem to be keeping this disease at bay. Another kind of question that we wanted to get at immediately is at what time, just in a way to try and identify peak periods of risk for infection. And the way that we went about this is we wanted to figure out when spores of Monolinia rubi are floating around in the air. And if there's spores in the air and there's feasibly susceptible tissue, then you could see how that would be a period of high risk for infection. And so the way that we went about this is we deployed a series of these rotating rod spore samplers throughout Northern Washington. They're very simple, machines. They're constructed from PVC pipe. And we deployed these to 2 different raspberry fields up in Linden, Washington, in collaboration with Lisa Jones. And if you look closely here, and I think I can click it, and a video might start. But what's going on here is this. There it goes. It rotates really fast, and there's 2 spore rods that are lined with plumber's grease. And as they spin, any spores that are floating around the air and even small insects that come into contact with the rods will get stuck to them. And we switch these out every 3 or 4 days over the course of the entire growing season. And that gives us 3 to 4 day snapshots of what spores were floating around in the air at that given time. And so we do this over the course of the entire growing season, and we generate a 3 to 4 day collective snapshot of what spores are floating around in these raspberry yards, these raspberry fields up in Linden, Washington. And once we have all these rods, we bring them back into the lab, and very similar to a PCR-based COVID test, we do DNA extractions on the spore rods, and then we have a PCR-based assay that is designed to detect Monolinea species. Currently, the only one available to us is only good enough to get us down to the genus level Monolinea, so that wouldn't differentiate between Monolinea rubi, our pathogen of interest, and Monolinea laxa or Monolinea vaccinii corumbosi, which those pathogens are floating around up there too. And so something that I've been working on this winter has been to recreate this PCR assay, but make it sensitive and specific enough to only detect Monolinea rubi, so that we know exactly, that it's Monolinea rubi spores in the air and not spores from some of these other closely related pathogens. And so that has been working really well. And so we have 6 candidate PCR tests that are all working. We're working on just a little bit more validation, but that's something that we're doing this winter. We're going to repeat this project a couple more years. And the ultimate goal is, if we find peaks in when spores are in the air in these raspberry plots, we are looking to either associate that with calendar days and time of year, or there's potential that it's associated with weather events, and we might be able to, if it's consistent enough across years, start to generate some predictive capabilities associated with when spores are released into the air. Yeah, that is all I had as far as other things going on in the lab. We've submitted 4 samples for complete genome sequencing, and that'll give us a really in-depth understanding of kind of the genetic diversity of our pathogen, and further enable us to develop more of these molecular PCR-based and qPCR-based tools. Yeah, that's about it. And so I'm happy to take any questions that anybody might have from the in-person audience or here online. Cool, Inga.
I missed the beginning of your talk. Did you mention how widespread Monolinea rubi is?
So we haven't done any so the question from Inga was how widespread is Monolinea rubi up in northern Washington and British Columbia. So as far as literature goes in the United States and Canada, that's the only region that it's ever been reported. And recently, we have not done any exhaustive sampling, in a kind of an organized way where that's probably something that we're going to get going this summer. But at this point, we haven't even seen it in too many commercial yards. So it's possible that, application of some of these conventional fungicides is keeping the disease at bay pretty well. A lot of our samples either come from breeding plots that don't have any sort of chemical applications to them. Other cane fruit that are growing in, just around the plots that grow wildly. And then occasionally in organic plots where, again, you're not having those conventional fungicide applications. You can see some prevalence of it as well. But it's been pretty well documented that it's hanging around at low levels, and it has that potential to kind of cause that significant yield reduction. But it seems like since the advent of some of these synthetic chemistries, it's been doing a pretty good job of keeping it at bay. But it's definitely worth kind of revisiting the pathogens, ecology and just very low level pathogen biology to have an initial game plan should acquisition of fungicide resistance happen in the population or anything like that come up, just to have a game plan to go. Sure.
You mentioned other key spots.
Yeah.
Could you expand on that?
Yes. Yep. So the question is, I mentioned other, cane fruit aside from raspberry. Where we had the spore traps deployed are in raspberry plots, but we've collected them from Columbia Star as well. So blackberry, and then the initial reports in 1959, those were on loganberry and thimbleberry as well. So its infection capability seems to be kind of widely distributed across the rubus family. Yeah.
Has it been found in Oregon, or has it been like
We've been looking, like even casually when we go for hikes and whatnot. I mean, there's cane fruit all over the place, and we've brought samples that kind of, like I alluded to at the start, the symptomology where it has that little bit of necrosis coming out of the fruit and just kind of that mummified fruit. There's a lot of physiological responses that can sort of mimic that. And so we've had instances where we're like, oh, this could be it, but we brought it in and haven't been able to isolate anything in Oregon as of right now. So as far as we know, everything that we've been able to collect has been Northern Washington, and then the original ports were out of British Columbia. Sure, go ahead, Inga.
Evidence from other monolinia of your expectation of resistant coloring within monolinia rubi?
It's tough to say. So the question is based on fungicide resistance type research in other monolinia species, could we kind of have any initial expectation of where resistance might occur? And it's tough to say because most of the evidence most of the research associated with acquisition of fungicide resistance has been done on the brown rotting monolinia. And there is evidence of resistance to azole fungicides. A lot of that research is coming out of the northeast and the southwest, but not as much for your mummifying monolinia. So I don't know if the expectation would hold true until maybe we get to take a peek at the genomes and get to look at kind of the similarity of those gene targets of the fungicides to see if kind of the motifs associated with the resistance in the brown rotting monolinia are also present in the mummifying monolinia rubi and other mummifying monolinia species. Cool. Well, yeah, I appreciate your guys' time, and I'm happy to talk or answer any other questions or anything else going on about the Stockwell Lab for sure as well.
Nitrogen. So some of you may know this, the Pacific Northwest has been a leader on researching and generating recommendations on nutrient management for both conventional and organic blueberries, in large part due to Dr. Bernadine Strick's leadership at Oregon State University. However, the experiments that provided the foundation for current recommendations were done in maritime Western Oregon. These conditions are extremely different from the conditions of organic blueberry production in Washington and Oregon east of the Cascade Mountain Range. In this environment, native pH of soils is high, and it can be anywhere from 7.8 and above. Growers do acidify the soils to make them suitable for blueberry production, but high levels of soil carbonites as well as alkaline irrigation water can buffer and elevate soil pH respectively, and growers do report this happening. In addition, soil organic matter levels are lower 1% and less is very common. Growers, again, do amend the soil using orchard wood chips to try to elevate soil organic matter levels. But add to all of that, we have warmer growing season temperatures and comparably higher levels of production. This really justifies the reevaluation and possible adaptation of nutrient management requirements for this unique but important production area. Just to give you a sense of the scale of importance, Washington State alone produced 35.4 million pounds of organic blueberry, and that was harvested from 2,700 acres in 2019, with most of that acreage in eastern Washington. This represented 46% of total national organic production with a value of $75 million. So it's not a trivial industry. So to continue to support this important and even growing industry, growers and crop consultants need data-driven recommendations on optimal nutrient management. And this is what the current project aspires to do, is to meet that need. So you see the objectives here on the right. I'm going to be focusing just on the first one. And that objective is to evaluate the effects of available organic fertilizer nitrogen sources and rates on blueberry plant growth, development, yields, fresh berry quality, soil chemistry, as well within the unique production region of eastern Washington. And for brevity, I'm just going to go over some of those data. But before that, I want to make sure I tell you a bit about the design of the study. So a replicated field experiment was established in a 4-year-old commercial and certified organic Duke field in Prosser, Washington. And this is an image of the field that you're looking at. The experimental design was a split plot randomized complete block design with a total of 4 replications. Our main plot was fertilizer source, and that was blood meal, fish emulsion, Wise Organic, which is a liquid fertilizer made from digested food materials and byproducts. And then our fourth treatment was a combination with 40% blood meal and 60% Wise Organic liquid fertilizer. The subplot factor was fertilizer rate within source, and that was at 50, 100, and 150 pounds per acre of nitrogen. And all of these treatments were decided upon based on feedback from growers within the region. They felt these were representative fertilizer sources and rates for their enterprises. Now, for this final project presentation, I'm just going to be focusing on data collected from 2018 to 2020. But I do want to acknowledge that we received funding from the Northwest Center for Small Fruits Research to continue this study, and the project will continue through 2023. I'm just going to highlight what I think are the most interesting and important results. So first, let's look at yield. For yield, we observed no differences in yield across our fertilizer treatments during the first 2 years. However, yield was slightly lower in 2020, and there was a trend for decreased yield in treatments containing high rates of our Wise Organic liquid fertilizer. Now, this decrease in yield with Wise Organic is not statistically significant, but that decline in yield may be attributed to the higher electroconductivity levels in Wise Organic. And this was something we were able to confirm with soil tests as well as test of the fertilizer. EC levels were simply higher, and that's been known to be problematic for blueberry that is sensitive to salts in the root system. Fruit quality variables were also impacted by yearly environmental conditions, and they were only marginally impacted by our treatments, specifically blood meal rate treatments, with firmness being highest in our medium and high nitrogen rate treatments, and titratable acidity being highest in the low nitrogen rate treatment. And I'm not showing fruit quality data, but I wanted to give you a summary of that. Now, leaf tissue nutrients, I'm only showing potassium here, and I will address that in a moment. What I first want to tell you about is nitrogen. So total nitrogen increased with rate except for our blood meal and fish emulsion treatments, but all of the tissue nutrients were within the recommended sufficiency ranges. In addition, all other nutrients were within their sufficiency ranges with the exception of potassium, magnesium, and copper. Potassium only showed year effects, which you're looking at here, and it continued to increase in concentration as the study progressed. Now, the lack of a treatment response for potassium is unsurprising to us because we did supplement both potassium and phosphorus throughout the duration of the 3 years of this study to standardize their rates across our plot so that we were only varying our nitrogen rate and our nitrogen source. Now, these results suggest that tissue potassium could be problematic in these fields because they were getting excessive according to our standards, and because we know that blueberry is sensitive to high levels of soil potassium. This is known, Dr. Strick has shown this. High levels of potassium is also likely responsible for the overall yield reduction that we observed in 2020. And we have since stopped applying additional phosphorus and potassium fertilizers. So next, I want to tell you about our soils data, specifically ammonium and nitrate. We measured this only in our 100 pounds of nitrogen rate treatment across all fertilizer sources. And we use these ion exchange resin membranes called plant root simulator probes. I'm just showing data from 2020, but 2028, 2019, and 2020 are all very similar. What I want you to notice is how low ammonium is in these soils relative to nitrate, which we found surprising. Now, this could be potentially problematic or concerning. So blueberry uses ammonium in its metabolism, and it lacks the ability to reduce and metabolize nitrate outside of its roots. The low ammonium could indicate a number of factors, such as root uptake. However, based on the literature and our understanding of these soils as well, it is likely that ammonium is rapidly being nitrified to nitrate due to the presence of nitrifying bacteria that are persisting in soil microsites. Now, nitrification is impacted by soil pH, and these are from a native soil pH level high in pH, but our average soil pH around the time that the first 3 years of the study was ending was an average of 5.5. Now, increasing soil pH can increase nitrification with maximum nitrification between a pH of 6.5 and 8.5, but we're not at a high enough level where we think that is contributing to the high levels of nitrate. So let's wrap up. So to date, our findings suggest that established Duke may be less sensitive to fertilizer nitrogen source and rate under the conditions of this study. In turn, this indicates growers could apply fertilizer nitrogen at lower rates, such as our 50 pounds of nitrogen per acre rate, because again, we didn't see a rate effect. And this is going to lead to no declines in yield and fruit quality based again on the conditions of our study in Eastern Washington. Lower rates of nitrogen can also reduce the potential for environmental pollution from excess nutrients, and there has been some work to show that high rates of nitrogen can also be problematic for certain cultivars of blueberry. Now, the lack of a fertilizer source treatment response in our study might be due and is likely due in part to us making potassium and phosphorus consistent across our treatments. And so there may be still a fertilizer source effect that we might detect as this trial continues, because again, we've stopped applying potassium and phosphorus. Still, high tissue potassium and knowledge that high levels of tissue potassium is associated with lower yields should serve as a cautionary warning for growers that are utilizing potassium-containing fertilizers in blueberry. Now, the high nitrate levels, as I mentioned, this was surprising for us, but it is likely due to the presence of nitrifying microbial communities that are persisting in the soil despite the lower soil pH. And this has been observed in other systems. But it suggests that to us, fertilizers that are slow to mineralize or nitrify might be more suitable for this production reason. So ammonium remains present longer and avoids that rapid nitrification. So as the project continues, we will proceed with data collection and information dissemination. The next phase of the project will measure net mineralization rates of the organic fertilizers that we're using in this study. However, we're doing it in a laboratory incubation protocol under field representative moisture and temperature conditions. And this is work that's being done by Dr. Gabe Luhu, who is a soil scientist here at Washington State University. And he has already found some pretty interesting findings that I look forward to sharing at future events. So with that, I want to thank you for your attention. A big thank you to our project team, the Northwest Center for Small Fruits for funding this work, as well as the other organizations that have contributed to our project. So I'm going to stop screen sharing, and my understanding is Q&A will be coming through the chat. Man, if you're thinking about questions, I'd be happy to talk about what we've observed in 2021, particularly in the incubation study, if anyone's interested in that. Okay. Oh, Carolyn wanted to know. So what we observed, well, first, the moisture conditions that we were trying to achieve, they were really wet. So the mineralization protocol, incubation protocol did suggest we're going to have to lower the moisture rates, but we saw very high denitrification rates, particularly in our Wise Organic treatment. We saw less denitrification in our blood meal treatment. And so we're starting to think more about, the conditions of the environment where these plants are grown, the fertilizers that are being grown, and denitrification. And that's something that as Dr. LaHoo will move forward with the study, he will be trying to capture that a little bit more to get a sense of denitrification potential. Yeah, but again, it kind of going back, it's to me and to our project team, it's suggesting that with rapid nitrification and then reduced denitrification of blood meal, this might be one of the more suitable fertilizer sources given the rapid nitrification that we're observing in these systems. It would be really interesting to measure the presence of nitrifying bacteria in the soil, but we're not there yet to confirm their presence. But another student working with, I think this was maize, not a berry crop, but they did confirm that there are nitrifying bacteria that persist in these soils. Thanks, Carolyn, for the interest. Yeah, and I'll also add, since we're starting to just look at our 2021 data, or we've been looking at it, we're not seeing a yield response still. I still haven't gone through all of the tissue nutrient data. I'm hoping to see potassium decline given we stopped supplying additional potassium, but when it comes to yield, we're still not seeing a yield response, which is now not really unheard of. We've got other collaborators here in Washington. I've heard of studies in British Columbia, and I'm aware of studies in Oregon where established plants with different fertilizer and nitrogen rates are not showing strong or any yield effects over the course of multiple years when varying nitrogen rate, which is interesting and is likely due to the plant's ability to compensate. Yeah, I welcome any other questions. There's a lot of great research happening in this part of the Pacific Northwest and for the organic blueberry industry. Dr. Scott Lucas has also been doing some really great work in collaboration with Dr. David Bryla on organic fertilizer sources and acidification technologies. Great. So I'm not sure if you're able to see, but Dr. Bernardine-Strick mentioned we have found higher yield with lower rates from establishment through maturity. So that's a great point. These plants were not in the establishment phase. I would consider them established. And I would agree and looked at your studies, Dr. Strick. There have been lower or higher yield associated with lower rates, but we're just not seeing that for the plantings that we've been working at for this study. And in other studies, I've read this as well. So interesting finding when it comes through established mature plants. And I guess I'll just kind of add to that too. You might have seen this presentation, Dr. Gabe LaHoo. He did a study for 3 years looking at different fertilizer nitrogen rates at 4 different farms in northwestern Washington. These are not certified organic fields, but they were plantings that were 6 years or older. And likewise, looking at low, medium, and high rates, approximately 30, 50, and 70 pounds of nitrogen per acre across various types of soil organic matter. No yield response has been observed as well over the course of the 3 years. Well, it looks like there's not a lot of questions remaining. So as we wrap up, just want to thank you for your attention. Really wish I could be there in person. Looking forward to resuming in-person meetings more regularly and interacting with the Northwest Center for Small Fruits Research community. All right, well, if you're attending, have a great evening. I think there is a dinner planned. If not, regardless, have a great evening.
You're good to go whenever you're ready.
Oh, okay. So, good afternoon, and I'd like to thank the organizers for making this a hybrid meeting so we can present remotely. So this afternoon, I'm talking about a project on recurring blueberry shock disease. And this is a project between Yanis Tanatakis' lab in Arkansas and myself. And in Yanis's lab, Dan Villamore is the person that did the high throughput sequencing and the cloning to make full length clones. Ava Waite and Daisy Stainton did the testing and cloning of the amplicons. And Tom Walters and Jason Myers are crop consultants here in the Northwest, in Oregon and Washington, boots on the ground. They know the fields, and they were able to help us collect samples and send them to Yanis and myself. So those are the players. A little bit of background. So the growers have expressed concerns about recurring shock disease in Washington and Oregon. And when we tested these plants for known viruses, in most cases, we only detected shock. Some of them had scorch, but that wasn't a big surprise. So that's where we're coming from. And then one of the resources we're going to make use of in the talk in this work was in 2015 to 2018, Yanis and I did a survey for blueberry viruses across the country, where we had either collected or in most cases people sent us plant material, and we extracted nucleic acid from 2,500 samples, and we tested those for 13 different viruses. But at the time, what we did was we took those extracts and saved them at minus 80 C, so we'd be able to do a survey in the future if we found any new blueberry viruses. So now we can do that survey out of our freezer. Well, the future's arrived, and we made use of that in this project. And the other thing to mention is that I retired at the end of 2019, right about the time when we found these 2 new viruses. And I knew it would take at least a year to get my replacement. So that's why I worked with Yanis. Most of the work was going to be done in his lab, so we could get a start on this recurring shock before the USDA was able to fill the position. And now the position's been filled. Dr. Dmitry Molev started in that position in May. So he's now leading this and other small fruit virology work for the USDA in Corvallis. And if you had questions, I strongly encourage you to go pound on his door or rattle his phone. So where did we find these viruses? Initially, we found the viruses in a project that Yanis and I did, funded by NCPN, where we were looking at using high throughput sequencing as a detection method and comparing that to PCR and ELISA for the really top tier plant in a certification scheme, because that's a really valuable plant that might be propagated into millions. But during that process, we found these 2 viruses in plants that we had as our standard, some of our standard controls, but we didn't know they were there because we didn't have specific tests. HTS looks for everything that's there, even if you don't, whether you know about it or not. So that's where we initially found those 2 viruses. And then working with Jason and Tom, we collected some samples from Oregon and Washington that were infected with, that had symptoms of recurring shock. And we did a few here, and in Arkansas, they did some as well. And out of 14 samples that we analyzed by HTS, high throughput sequencing, 13 of those had the ludiovirus and the blueberry shock. And we also tested 2 plants that did not have, that had typical shock. So that's another source of the ludios, is those field plants. And then we also, in the past, had worked on blueberry bronze leaf curl in blueberry. It's a disease in the Midwest, Michigan, Indiana, Wisconsin. And we examined one of those by HTS, and we also found the ludiovirus in material from Michigan. This is just a picture of a Draper field here in Oregon. And these plants are infected with blueberry shock, blueberry scorch, and the ludiovirus. And when you see this in the field, it looks like it should be a nematode transmitted virus because it's a very tight oval patch. But we had collected soil. Inga did not find any nematodes, and we did not detect any tobacco ring spot or tomato ring spot or peach rosette mosaic virus in these samples. These are some pictures that got from Tom Walters from material that he collected that were showing shock symptoms in subsequent years from their initial infection. And these were infected with the ludiovirus and blueberry shock. This is a picture of bronze leaf curl disease from Michigan that I got from Annemiek Schilder when she was there. And when we worked on this about 10 years ago, we found the BVA. But during that survey we did in 2015 to 18, we then realized that there were a lot of samples in Wisconsin, Michigan, Indiana that had BVA but were symptomless. So we knew that the bronze leaf curl was either caused by something else or a mixed infection of something with BVA. So plants exhibiting repeat shock symptoms in Oregon and Washington. So the HTS data showed that 13 of 14 had the ludiovirus. Several plants also had scorch, which was not a big surprise. But the ludiovirus was not present in plants that exhibit typical shock symptoms. So they had shock, and the following year they had recovered. So in those plants, we only found blueberry shock. And then once we had that work done, we developed one and had primers. We confirmed the presence of blueberry shock and the ludiovirus in those plants. And 13 of 14, the same 13, were positive in RT-PCR as were positive in HTS. And again, there were several positive for blueberry scorch. But the fact that we had a plant that was negative in HTS either suggests that the nucleic acid extraction was inferior, had problems, or that there's something else non-viral that can cause that type of symptom. So I talked about doing a national survey, and from the survey material we had out of our freezer, we pulled 1,000 RNA samples that had been stored at -80, sent those off to Arkansas, to Yanis's lab, and they started testing those for the luteovirus. And once we had primers for the new Carlavirus, they started testing that as well. The primers were based on sequence from the first 7 luteovirus isolates that they had found in their high throughput sequencing work in Arkansas. And Dan Villamore had generated full-length clones for those 7. So they, based on those 7, they developed primers that at least work for those 7. And since then, they've analyzed over 600 samples and found that the luteovirus is really very widespread. And given that the primers were based only on 7 sequences, the incidence that we're going to talk about in a minute is probably underreported. It's probably higher than that because with only 7 sequences to work with, and if we look at the diversity of the virus, there's a good chance that we're missing some of the isolates. So the occurrence, it's very widespread. Out of 607 samples, when we put this slide together, 316 were positive for the luteovirus, so more than 50%. And the variation is quite wide. So on that 83%, out of 100 nucleotides, only 83 of them match. So there's a lot of mismatches in the sequence. You hear about the COVID-19. It's a virus that mutates a lot. RNA viruses tend to do that. This is an RNA virus, so it's not at all surprising. And like I said, the samples we collected and started to work on are from the Northwest, Michigan, and New Jersey, which are 3 major blueberry production areas, but also 3 of the older blueberry production areas in the US. And you can see in the Northwest, we had almost 30% of the plants we looked at were positive. But in Michigan and New Jersey, it was over 60%. And this is an example of the PCR. That bright band is about 1,000 bases. And so that's what they cut out and cloned and sequenced. And that's the 316 sequences they now have and that they're working to develop better primers. And so also once we had the detection method, we were contacted by Kim Hummer at the repository because we had talked about this at some meetings, and she was worried that they could have that in their material. And she identified 16 cultivars that are commonly distributed or most commonly requested from the repository, and we tested those for the luteovirus. And 7 out of those 16 were positive for the luteovirus, and all were symptomless. So it's widespread, and when it's by itself, at least, it's symptomless. And then, like I mentioned, we had the new Carlavirus, and here I put up the phylogenetic tree because you can see it's most closely related to blueberry scorch. But it's distinct enough that it would be considered a different virus. And, whoops, I went the wrong way in that one. So with the novel Carlavirus, in ELISA test, it gives a very weak reaction, 0.2. Those infected with blueberry scorch should be between 2.0 and 3.0. So this is about 1/10 or less than 1/10 of what we would expect. And I think in a lot of cases, those would probably be called negative. So they'd give you a false negative if you wanted to call that blueberry scorch. And also the PCR test that we had for scorch did not work with this virus. And when we put blueberry scorch virus into Nicotiana occidentalis, we get very mild symptoms. But in this case, with the new Carlavirus, we get very severe symptoms. So this is with the new Carlavirus. And in quinoa, blueberry scorch causes symptoms. The new Carlavirus doesn't cause any symptoms. So there's difference biologically in herbaceous host, and we don't know if there's going to be differences in blueberry. But we are in the process of purifying that. Hopefully by this next growing season, we'll have antibodies for this particular virus that we can test for. And also in the survey on this one, the incidence is about 4%. The luteal virus was over 50. So this is much less common. Again, the identity, it's quite variable. And we only had 20 partial sequences that we were working with. So we don't have as much information on it, but we did find it in the Pacific Northwest, in Michigan and New Jersey. And like I said, about 4%, 20 out of the little over 500 samples that have been tested. So the novel luteal virus is widespread. It's present in most of the blueberry plants that exhibited recurring shock symptoms. It was present in the one plant of bronze leaf curl that we tested. That's just shows us it's there. It doesn't really tell us a lot about interactions. Widespread in the major producing areas for northern high bush blueberry in the U.S. It's also present in British Columbia. Present in significant number of cultivars at the clonal germplasm repository. And some of these are old cultivars that have been around a long time. And some of them are much newer germplasm. And I would say it's very likely that this virus now occurs wherever blueberries are grown worldwide. This virus, best guess, it's been around a long time because there's so much variation. And, New Zealand is probably the most difficult place to ship plants into. But if we ship blueberry plants to New Zealand or Peru or wherever, and they test them, they're going to use all the known tests, and they would have been negative. So they would have let them into the country. So I'm pretty confident that this virus probably occurs wherever we grow high bush blueberries. But also there are asymptomatic plants that were positive for both the luteal and the shock. So there's something else happening that we don't understand, and that still needs more work. And given that, we do not know that the luteal virus is a player in any blueberry disease at this point, but that's something that needs to be studied because it's a virus that we can clean up, and it'd be a good idea to know how important it is in production. So what are the next steps? So use the 250 plus sequences we have to develop better primers, and that's being done by Yanis's group, and doing the similar thing with the Carla virus, and that's being done also with Yanis's group and funded through the Farm Bill. Cultivar differences to infection by these two viruses, single or mixed. TBF is to be funded. Identify vectors for the luteal virus and Carla virus. If we're going to do any management, we need to know how these things are moving in the field. Virus vector interactions. How long does the virus persist in the aphid or the insect, most likely insect, when it's picked up? Are there reservoirs out there other than blueberries where it might be? How do we manage those vectors? Examine recurring shock disease, looking at different combinations. And then the big thing is to examine symptomatic plants that are negative for blueberry shock and blueberry scorch and the luteal. This might be just a factor of the primers that were being used are not adequate. One thing that we probably should be looking at is blueberry shock virus itself. Is there a variant, a Delta variant, Omni variant, or Zeta variant, whatever you want to call it, that might actually cause severe strain that would cause recurring shock symptoms, even there's nothing else there? And you notice all of those last set are to be funded, so you could pick and choose what you'd like to put your money towards. And then long-term, look for sources of resistance, tolerance, or field resistance. And this is continue to be funded because, the commissions have been funding that for a number of years now with Chad, and hopefully continue that with Claire. So that's all I have on what we've done. A big thank you to the funding sources, the Northwest Center, the Washington and Oregon Blueberry Commissions, the Farm Bill, and the National Clean Plant Network. And, so I'm retired and I'm looking for other research projects. And, they tell me that the meaning of life can be discovered in a fine bottle of wine. I haven't found that bottle yet, but I'm enjoying the research. And this one is to be funded. So I'm looking for people interested in funding that one as well. And with that, I'll finish and just wish everybody a happy holiday and great 2022. And take any questions. Any questions? The best guess for vectors on both of these, on the luteovirus and the carlavirus, would be aphids. All the known luteoviruses are transmitted by aphids, but we need to figure out which aphid and then study the aphid biology. And the same thing with the carlavirus. Best guess is going to be an aphid. Too bad it's not nematode vectored. Well, if you look at the field, especially that one field, it looks like it should be nematode vectored. It's the classic oval patch in the field like you would expect with a nematode. Okay, if there are no questions, hope the people that are going to dinner enjoy the meal. It's a great spot. I think I can quit sharing screen. I don't see any questions, so